This method can help answer key questions in the RNA biology field, such as what is the biological significance of RdRp or TERT in human cells.
The main advantage of this technique is that this assay is established as the first sensitive method to detect human RdRp activity expressed in cells.
Begin this procedure with preparation of HeLa cells as described in the text protocol. Wash the cells once with 10 milliliters of PBS. Then, centrifuge the sample at 1,450 times gravity for 5 minutes at 4 degrees Celsius and discard the supernatant. Use 1 milliliter of ice-cold lysis buffer A per 10 million cells to lyse the cells by gentle pipetting. Sonicate the sample in a 1.5 milliliter tube at a sonicator amplification of 25% for 10 seconds. Following sonication, centrifuge the sample at 20,400 times gravity for 15 minutes at 4 degrees Celsius. Collect the supernatant and transfer 1 milliliter each of the supernatant into 1.5 milliliter microcentrifuge tubes. Pre-clear the lysate by adding 40 microliters of pre-washed protein A agarose beads per 1 milliliter of the lysate. Mix well by inverting the tube several times. Then, rotate the sample for 30 minutes at 4 degrees Celsius. Centrifuge the sample at 13,000 times gravity for